Now the first transducer mechanism is G protein coupled receptors, also known as GPCRs. Now GPCRs, these are cell membrane receptors which are linked to the effector. Effector can be any ion channel, any carrier protein or enzymes. And these are known as G protein coupled receptors because they are coupled with GTP activated proteins, that is guanylyl triphosphate activated proteins. As far as the structural organization is concerned, there are 7 alpha helical membrane spanning units. These are the amino acid segments, and there are 7 of them, which are alpha helical segments. And on the cytosolic side, they are coupled with G protein units, whereas on the extracellular side, there is a agonist binding site. Now the G proteins which are on the cytosolic side, they are heterotrimeric in nature, heterotrimeric in composition. That means there are 3 subunits, that is alpha, beta and gamma, and in the inactive state they will be attached to GDP. Now what will happen when an agonist will come and bind to the receptor? When an agonist binds to the receptor, now these proteins, they are activated by GTP. Now GTP will come and knock out GDP and replace GDP in this unit. After that, on GTP binding, the alpha subunit that will dissociate from the trimer, and a beta gamma dimer will be dissociated. Now this activated alpha subunit, that goes and it either activates or inhibits the effector, which can be any enzyme or ion channel or carrier protein. On the basis of this alpha subunit, the G proteins, they can be classified into 4 categories, that is GS, GI, G0 or GO and GQ. And all of these 4 categories have different functions. For example, GS proteins, they are responsible for the activation of adenylyl cyclase, and also they are responsible for calcium channel opening, whereas GI is for inhibition. They inhibit adenylyl cyclase and they are responsible for the opening of potassium channels. GO is for calcium channel inhibition, and GQ, that is responsible for the activation of phospholipase C. Now there are 3 major effector pathways of GPCRs. As we know, adenylyl cyclase activation is done by the GS proteins. So let us consider now these proteins are GS proteins, and our agonist, let us suppose it is adrenaline. Now when adrenaline will come and bind to these GPCRs, what will happen? The GDP will be replaced by GTP, and there will be dissociation of this trimer. Now this activated alpha subunit, it goes and affects the effector, which is adenylyl cyclase in this case. Now adenylyl cyclase, when it will be activated, it will start converting ATP into cyclic AMP. That is why this pathway is also known as cyclic AMP pathway. Now cyclic AMP is our second messenger. It directly stimulates the calcium channels also, and it functions through cyclic AMP dependent protein kinase A. Now protein kinase A, when it is activated by cyclic AMP, it alters the functions of certain enzymes, ion channels, and carrier proteins, and thus it produces response like increased contractility or impulse generation, glycogenolysis, lipolysis, etc. And opposite effects are observed in case of GI proteins, which will cause cyclic AMP inhibition. They will inhibit adenylyl cyclase. This will inhibit adenylyl cyclase, and there will be no formation of cyclic AMP, and all the functions which were to be produced, they will be inhibited. Now let us discuss the next pathway, which is phospholipase C or IP3 DAG pathway, which is carried out by GQ proteins. In this case, the agonist will be histamine. Now, what happens when histamine comes and binds to the GPCRs? Again, this heterotrimeric unit, it will be activated by GTP, and these GTP activated proteins, they will dissociate. The activated alpha subunit, now it activates phospholipase C, which is the effector in this case. Now phospholipase C, it will start converting membrane phospholipids, that is PIP2, which is phosphatidyl inositol 4,5 bisphosphate. It will convert PIP2 to DAG and IP3. IP3 is inositol 1,4,5 triphosphate, and DAG is diacyl glycerol. Now DAG, it remains in the membrane and it activates protein kinase C with the help of calcium. And IP3, this is water soluble, and being water soluble, it diffuses to the cytosol, and in the cytoplasm it reaches the endoplasmic reticulum, and it mobilizes calcium, or it releases calcium from the endoplasmic reticulum. And we know when the calcium level increases in the cell, the contraction will be increased. So this was the IP3 DAG pathway. Now the third pathway in GPCRs is channel regulation. In channel regulation, what happens is when the G proteins, they get activated, they can directly open or inhibit the ionic channels. For example, the calcium and the potassium channels. They can directly either they can open or inhibit these channels without the use of second messengers like CAM or IP3. They do not use these second messengers. They directly act on these ionic channels. For example, the GS proteins, they act on the calcium channels. They open the calcium channels, whereas GI and GO proteins, they open the potassium channels. So they can bring about depolarization, repolarization, or hyperpolarization of the cell. And these opening and closing of the cell, they can be they are the function of beta gamma dimer, because alpha unit was involved in the production of second messenger. Now this dissociated beta gamma dimer, it goes directly to the ionic channel and either opens or inhibits this. So this is the third pathway in case of GPCRs. Now the second type of transducer mechanism is ion channel receptors, which are also known as ligand gated ion channels. Now as far as the structure is concerned, it is pentameric in nature. That means it will be having 5 subunits, and they have 2 of the alpha subunits. They have beta, gamma, and delta subunit, and these 2 alpha subunits, they have the agonist binding site on the extracellular site. Now, these are the cell surface receptors and they enclose ion selective channel. That means they are specific for a particular ion. If these ion channels are for sodium ions, they will only allow the passage of sodium ions through them. So every channel is specific for their specific ion. For example, sodium, potassium, chloride, or calcium. So they are specific for their specific ion. Now what happens is, for example, this is our ion channel and it is lying in the closed state now. And when it binds with the agonist, what happens? Agonist binding or ligand binding, that will lead to the opening of this ion channel. Now this ion channel, it opens and it allows the passage of the ion through them. Thus, the opening and closing, that only depends upon the binding of the ligand. No involvement of coupling proteins or second messengers. Therefore, this transducer mechanism is the fastest mechanism for drug action, which acts in milliseconds. The third type of transducer mechanism is transmembrane enzyme linked receptors. Transmembrane because these receptors, they pass through the whole of the membrane. Enzyme linked because the cytosolic site is linked with an enzyme. So let us discuss the structure first. The extracellular part is the ligand binding site, and they mostly bind with the peptide hormones such as the insulin or the nerve growth factor or epidermal growth factor. And this is the transmembrane helical peptide, coiled, therefore it is known as helical. And the intracellular subunit, it is having the enzymatic property, and generally commonly it is having the protein kinase property that phosphorylates tyrosine residues. Now what happens is, in the unliganded state, they remain as a monomer. Only single unit exists. But what happens when there will be a hormone binding here? For example, insulin binding or nerve growth factor binding. Then what happens is, hormone binding induces dimerization of the receptor molecules. They will form a dimer, and there will be conformational changes which will activate the protein kinase in this cytosolic site, and there will be phosphorylation of these tyrosine residues. Now the phosphorylation of these tyrosine residues, they will increase the affinity of these enzymes for substrates, especially for proteins which are having SH2 domain. So what will happen? This phosphorylation will activate these proteins, and phosphorylation of these proteins will take place. Now they are phosphorylated, and we know when proteins get phosphorylated, they are activated, and hence they will produce a response. And that response in this case can be, there will be activation of metabolic reactions, there will be cell differentiation, cell growth, etc. Next type of transducer mechanism is JAK-STAT binding receptors. They look somewhat similar to the transmembrane enzyme linked receptors, but the difference is they do not have the intrinsic kinase activity. Let us understand from the process itself. So when there will be agonist binding, similar to the previous one, this agonist binding will induce dimerization and conformational changes. And these conformational changes, they will increase the affinity for cytosolic tyrosine protein kinase, which are also known as JAK molecules, that is Janus kinases molecules. These are basically tyrosine protein kinase molecules. When agonist binds, now these cytosolic molecules, JAK molecules, their affinity is increased and they go and bind to the receptor. When they go and bind to the receptor, now the phosphorylation of the receptor takes place. The tyrosine residues, they get phosphorylated after the activation of JAK. Now phosphorylation of these tyrosine residues, that will increase the binding of the STAT molecules. Again, STAT molecules, they are freely moving protein molecules in the cytosol, which are known as signal transducer and activator of transcription. They go and now they bind with this JAK. When they bind with this JAK, the tyrosine residues of the STAT, they also get phosphorylated. After phosphorylation, these STAT molecules, they get activated and a dimer is formed. Dimer of STAT molecules is formed, which now dissociates from the receptor. When it dissociates from the receptor, this dimer translocates to the nucleus. It enters the nucleus. There it can regulate the gene transcription, and after the gene transcription, it can regulate or it can produce a response. There will be regulation of transcription, gene transcription, and there will be response. Now the fifth and the last type of transducer mechanism is receptor regulating gene expression. These are also known as glucocorticoid receptors. So the glucocorticoid receptor looks somewhat like this, which is attached to the HSP90, which are the complex proteins known as the heat shock proteins. So they are attached with these complex proteins, and on the carboxy terminus they have a binding site, and this is also known as the hinge region. After this we have a DNA binding site which is having two zinc fingers, and then we have a amino terminus. Now let's discuss the process directly. Now glucocorticoids are lipid soluble chemical messengers, so they can easily penetrate the cell and they bind to the cytoplasmic or the nuclear receptors, these proteins which are bounded with these heat shock proteins. After the binding of glucocorticoid, now these proteins, they are dissociated. After dissociation of these proteins, now these receptors, they can form a dimer. That means a similar structure like this, they join together and they form a dimer. After forming a dimer, they can go inside the nucleus, or if they are already in the nucleus, now they can bind to the DNA. When they bind to the DNA, what happens is they bind to a specific region which are known as GRE, that is glucocorticoid responsive elements. When they bind to this region, now they can regulate transcription. That means a particular mRNA will be formed. After formation of mRNA, it goes outside the nucleus and the translation occurs in the ribosomes and proteins will be formed. These particular proteins, they are responsible for the alteration of the cell's functions.